So today's lecture will be about sampling methods in research. Okay. So actually, about this week is mostly about how you are going to sample. You get your sample from a population. Okay. So in a way, sampling is the process of selecting a few, a sample here from the population. So you have a bigger group, which we call population, right? But it doesn't mean that it's the total population of one country, for example, unless that is what your research is about. Okay, so just to make it clear, so population is the bigger group, and then how you choose your sample, this smaller group, that is the sampling methods, right? So the basis of doing this is because you want to estimate or predict, something about the population. But because to do that for the whole population is sometimes very, very problematic, or it's very hard to do. Okay, so you choose a sample which is a smaller amount, so you can actually access them a little bit better. Okay, so sample is always a subgroup of the population of interest. Okay. So basically, we want to estimate what is happening in the population. So that is very important here, because you will see that this basically directs the aim of your sampling methods. All right. Now, okay, need to sample. Cool. Okay. So I don't know if you guys know about this, but right now, our government, the Malaysian government is doing a census. Okay, a census is done every 5 years usually. So census, you do the survey for the entire population of Malaysia, right? So census is basically the most representative way when you want to talk about a population. Because census, it's definitely going to assess all of the people, all of the members of the population. But a survey of the entire population is impracticable. Okay, so you, especially while you're doing research project, so you're not going to have enough time, enough budget to do a census. That's why we do sampling. Okay, because you can get access to a smaller portion of the population. Okay, but the questions here, right? How representative is the sample of the whole population? Okay, remember, because you're only choosing some members of the population. So how can you make sure that this small sample will represent the whole population? Okay, and then how similar are the characteristics? Okay, because always in whether it's sample or population, different characteristics. Okay, so if in your research project, there's always certain characteristics that you're looking at. Okay, and then how you choose your sample, how can you make sure that the characteristics are similar? Okay, similar and representative. So that is the keyword in here. Right. So just a quick highlight. So difference in population, in sampling. Okay, so populations, it does not necessarily mean a number of people. Okay, it's not necessarily means that. So it can also mean the total quantity of things that you are looking at. Okay, so not only, so we are talking about this not only for survey, okay, for those who are doing pure sciences, or because people always say surveys is for social science, all right? But for those who are doing experiments, for example, you do work with population and sample. Okay, so imagine if you are in agriculture, for example, just bear with me. So you are testing one sort of treatment to a plant, okay, to make that, okay, rice, for example, paddy, you want to make that rice more resistant to the climate change, okay? So you choose your sample as maybe 1 acre of paddy plantation, okay, and that will represent the whole sort of plantation in that region, okay? So in that sense, population does not only means people, so that is what I want to highlight, because of course, naturally, when we talk about population, you will think about people. They are alive human beings, but sometimes it doesn't always means that, okay? And not even objects, organizations, it can be events as well, right? So just keep that in mind. We have population and we also have sampling frame. So what is sampling frame? So within a population, okay, you will only look at specific groups. Okay, so you have, maybe here you have population of the residents in Selangor. Okay, that is your population. And then your sampling frame, maybe you want to look at waste segregation in Shah Alam. Okay, I know I always use Shah Alam as example. I'm not living in Shah Alam, by the way, but you know, that always comes to mind. So I don't know why. Right. So your the residents in Shah Alam, that is your sampling frame. Okay, and then you choose your sample. Okay, your subjects, the actual people who are living there, based on your sampling methodology. All right. So I hope that is clear. So in summary, yes, you have your population here. So you actually, your research want to talk about the population. You want to generalize something about the population, but you cannot access everyone in there. So you have your sampling process where you select your sample. Okay, and this sample is basically what you are focusing on your research. Okay, so your data, your basically the data analysis, data collection, everything is here at your sample. But because the sampling process takes into account the representativeness and also the generalization, so you can actually infer. Okay, you can infer your population to the results that you get in the sample. Okay, so I hope that is clear. So basically in design, sampling design process, okay, you always start with your population first. Okay, and you will determine the sampling frame and the sampling procedure. Okay, so here it is actually quite important, these 3 steps, okay, because this will determine whether you are doing probability sampling or non-probability sampling. Okay, more on that after this. But after you have decided on probability or non-probability, you will determine the appropriate sample size. So this is the number of samples that you need in your research. Okay, and lastly, you execute this sampling design. So you've got, if you're doing survey, you disseminate the survey. If you're doing experiments, you start your experiments. Okay, so that is the natural process of in sampling design. So basically, I've said to you about probability and non-probability sampling. Okay, so one thing to highlight is usually in quantitative research, you do probability sampling. Okay, because you see all the questions here, these are what quantitative researchers are thinking about. So you want to know the cause and effect of something, or you want to measure something. Okay, you want to generalize something, but you only have access to small groups. Okay, and you want to make sure others can repeat your findings, right? So in this case, probability sampling is the best approach, okay? Whereas if you are a qualitative researchers, okay, see the question. The questions are different. Okay, you want to see the world through the eyes of your respondent, okay, and you want to describe what is happening in a lot of detail, okay, and sometimes you want to see about social change, how it occurs, you know, and how certain people approach that, right? So in qualitative researchers' questions, non-probability sampling would be the best approach. Okay. So we can basically we can categorize this probability sampling and non-probability sampling into different methods. Okay, so we have here probability sampling methods. There's 4 types, and with non-probability sampling as well, we have 4 different types. Okay, but the basic thing is that you have to remember probability sampling. Okay, for quantitative researchers, it gives the most reliable representation of the whole population. Okay, so this is very important because for non-probability techniques, you cannot generalize. Okay, because the way non-probability sampling methods are done, they are not enough to, they do not fulfill the criteria to enable the results to be generalized. Okay, so if you remember last week, we talked about random, okay, sorry. We talk about, so the selection has to fulfill all those criteria before it can be used to generalize, okay, before it can represent the population, right? So in probability sampling, of course, it's the sample you choose from a larger population, okay? But the important thing is that it's based on the theory of probability, okay? Because what is important in probability sampling is it uses random selection, okay? So anyone or anything in the population has a known and equal chance of being selected, okay? So you see, you eliminate bias here. So you will, you basically, you want to know that you have your population, okay? So anyone there is has an equal chance, and that's why you can basically represent, so your sample can represent the whole population, okay? So I hope that is very important, because a lot of people forget about this when they are doing their sampling methods. Okay, so when you do your sampling methods, when you do your analysis, okay, you have to remember back on this, because probability, yes, then you can discuss, you can make generalization, you can conclude, okay, whatever findings that you get is something that the population also could exhibit, you know, the problem or the solution. Whereas if you are doing non-probability sampling, okay, you cannot discuss about generalizing that results, okay? That is the limitation. But qualitative researchers, they're not actually interested in generalizing. So you have to remember back on that. So you want to look in detail about a certain thing, right? Now, probability sampling uses statistical theory, okay? So you can actually eliminate any random errors or systematic errors because you are using statistics, right? And with this, your sample can predict that their responses will match the overall population. Okay, this is what we have been talking about. important issues in probability sampling is, again, representation and generalization. Okay. Of course, there's always error. Error is something that cannot be eliminated. But you are aware, and in probability sampling, you can actually quantify this. Okay, so you can be sure that the numbers that you get is correct and representative of the whole population. Okay, sampling error is the chance occurrence that the randomly selected sample is not representative. Okay, and this is due to the sampling techniques, which is why sampling, when you're doing the sampling methods, it's very important. Okay. Secondly is sampling bias. Of course, there's sometimes you are sometimes biased in creating the sampling design. Okay. So types of probability sampling, there's 4, remember just now. So there's simple random sampling, cluster sampling, systematic sampling, and stratified random sampling. Okay, so all these methods are random. So which is why it falls under probability sampling methods. So in simple random sampling, okay, it's completely random. So so each element, okay, each element in the population are given an equal opportunity to be selected. Okay, and the methods are, it's very simple. If you have, like here, 12 elements, okay, you have 12 elements and you want to do the sample, you choose 3. Okay, this is a generalization. Okay, so it's not, population cannot be 12, just 12, unless in certain cases. But what I want to say is you have 12 here, and then you can either use fishbowl draw. Okay, fishbowl draw is something that is similar to lottery. So you have 12 papers, for example, in one bowl, and you choose 3. Okay, so you actually got random numbers from there. Simulating is done through computer program. Okay, that is also available in SPSS. I'm not sure if you are familiar with SPSS. I think so, but yeah, a lot of you are probably going to use that for your analysis and sampling methods. Okay. And lastly, you have a table of randomly generated numbers, something like this. Okay, so I know that it seems like, doctor, this seems like so simple. Where is the scientific techniques here? So basically, because this is random, right? So it is used a lot, okay? But you have to remember, it means that you have access to all members of the population. Okay, so remember last week I was talking about if you want to send surveys. Okay, imagine if you are choosing one housing area, okay, one taman, for example, Taman Sri Serdang. So you must have the address of all of these people, okay, and then you assign numbers to these addresses, and then you do the random selection. Okay, that's how you do it. It's not just simply about 1, 2, 3, 4, 5, but actually it's the randomization that is the most important thing. Okay, so the advantage is minimal knowledge of population needed. Okay, and it's easy to analyze the data because you're sure that it's random. Okay. But the disadvantage is it's a low frequency of use. Okay, you do not use your expertise basically in selecting the sample. Okay, and there's a larger risk of random error. Okay, so that's for simple random sampling. Okay, next is stratified random sampling. Okay, so in this case, you have your random population here, and actually you have your own categorization, okay? Because you know there's the population always have different things. So the easiest one would be gender, okay? So 50%, maybe 50-50 or 60-40, right? So you can imagine you have two groups in one random population, if you're talking about gender, right? And you want to make sure that you have enough sample from these two groups, right? So that is why you do stratified random sampling. Okay, you don't want to, if you're only using simple random sampling, okay, you might end up with, instead of 60-40, you get something like 80-20%. Okay, and if your study is very related to gender, you can imagine why this is a problem, because your sample would not be representative of your population. Okay, so remember, I think Yap, you have asked about this a few weeks back, right? So yes, economic, you know, if you're looking at something at the social, the economy of your population, okay, you want to make sure that you exclude the, you know, the teenagers, okay, or the elderly from your population. So you can do something like this, or next we will look at cluster as well. But yeah, I would say that stratified random sampling is the way to go for that. Okay. So basically you have your random population and you have your groups, and then so here it has 4 groups. Okay, for example, please bear with me. What happens next is you choose randomly from each group. Okay, so you got, maybe you got one here, one. Okay, just an example. So in stratified random, you separate your population into groups, and you make sure that you have representative of each group. Okay, you're not only choosing one group. Okay, that is a different type of sampling. So this is an example. So if that one company, it has 1,000 employees, okay, and 800 are full-time employees and 200 are part-time. Okay, so in your research, you want to select 100 people. Okay, so based on that, okay, the same proportion. So you have you need 30 people from the full-time and 20 people from the part-time employees. But what's the most important is you have to make it random. So you already group the people, and then you have to choose them randomly until you get 100 total people. Okay, stratified random sample. Okay, so the advantages are it assures a representation of all groups in the sample population. Okay, and characteristics can be estimated and you can make comparisons. However, there's also disadvantage because you need to have accurate information, okay, on the proportion of each stratum. So sometimes this is quite difficult, okay, and which is why it is quite costly, right? But there's always, that's how you develop your methodology. You have to think about how you can access this sort of data, okay? If you are looking at something that is really related to a population, you can always use census data, okay? So you can actually get a general representation of whatever that you're looking at, okay? Age, gender, economic, even the household income, which is sometimes something that is a lot of people do as well. Okay. Next is cluster random sampling. Okay, this is a little bit different from stratified, although it is somewhat similar. Okay. So in this sort of sampling, you randomly select intact groups, okay, not individuals, right? So what is easiest to look at is locations, right? So basically, if you look at the image there, so you have your population here, okay? This is if you are only doing simple random sampling, okay? So you're just going to select all of those, your sample from the population. However, if you're looking at clusters, okay, you might want to look at the where the people are based at. Okay, maybe region 1, region 2, region 3, region 4, and you want to select only region 2. Okay, however, in this type of sampling, you are going to choose all of the members from that cluster. Okay, so that is where it's different from before, from stratified, because in stratified, you will choose randomly sample from each group. But for cluster random sampling, you're going to choose that particular group, right? So it can also be multi-stage cluster as well. So you can actually do a number of different stages. Okay, that is also possible. The advantage is it's very useful when population are large and spread over a large geographic region. Okay, it's convenient and expedient. It can estimate the characteristics of both cluster and population. Okay, because one thing I want to highlight is when you're doing sampling, especially in probability sampling, okay, you have your based on statistics, right? With enough number of sample, that sample actually is a normally distributed sample. Okay, so I hope you this is related to statistics as well. So which is why in normal distribution population, okay, in your sample, you can generalize all the findings. So if you want to look more into this, then you can search about the theory of this one, the normal distribution of sampling. Okay, so the disadvantage, of course, is quite costly because you will need to have access to all members of the group that you choose. Okay, however, it's always a matter of the middle ground between advantage and disadvantage. So your selection will depend on which is most advantageous to you and which type of sampling is the most suitable for your research questions, right? So this is just one something that I want to share, the difference between stratified and cluster, but I've already touched upon that while we are talking about it. So one thing I can highlight is the choice, okay? Choice of elements from within each group for stratified. So you do it randomly, okay? Selection of the elements. But for cluster, you do random choice of subgroups, okay? Because you remember you have to choose all elements in that groups. But the selection of that group is random. So that's where the random part of the sampling comes in. Okay. Now we have systematic sampling. This is the fourth one. Okay. So basically, you choose every nth individual to be a part of the sample. Okay. So for example, every fifth person is included in the sample, right? So it may seem very simple, but why is it systematic? Because you want to make sure that it's an extended implementation of the probability technique, okay? Because you select your sample at regular periods, okay? and there is an equal opportunity for every member to be selected, right? So one thing that I've seen people do this is, okay, very simple example is people at shopping complex. Okay, they wait at the entrance. So every third person, so 1, 2, 3, the third person, they will give the whatever that they want to do. I mean, it can be survey or it can be gifts. Okay, so it's how because you know you can't actually, this is quite random because you don't do people, you know, with timetables, for example. Okay, entrance into shopping malls, it is random, right? So this is one simple example of system, moderate cost and moderate usage, and it's easy to draw the sample, okay, and easy to verify as well. However, the disadvantage is periodic ordering is required, right? So it may be, depends on how you want to use the sampling method, okay? You might need to think about the timing, okay? How long is this going to take, right? And that will, you know, affect sometimes. It may introduce bias as well, okay? Now, this is, the previous 4 is about probability sampling, okay? Linking back towards quantitative sort of research. So for non-probability sampling, okay, you have to look at why you are doing this. Okay, because the selection of the participant usually relates to their characteristics and their knowledge. Okay, so the probability of each sample being selected is not known, because sometimes it's only based on your personal judgment or the convenience. Okay, so basically there is no statistical techniques to measure random sampling error because you are not doing this randomly. Okay. Right, there's 4 types, okay, 4 main techniques: convenience sampling, or also called purposive sampling. You have snowball sampling and quota sampling. Okay, so 4 types. We'll look into that one by one. For convenience sampling, it involves choosing respondents at the convenience of the researcher. Okay, so you can imagine if you are doing market survey. So market survey is something that I always use as an example because I think it's very general. So basically you, sorry. So it's based on your convenience, on the convenience of the researcher. Okay, so if you look at that, the sample that you get are the people that you have access to. So if you are in a shopping mall, you know, you approach people and those who have time to get back to you. So that is your sample. But you can imagine a lot of people also, you know, no, I don't want to be involved. Okay, that happens, all right? So that will not be included in your sample. Okay, but what's important here is that it's really, because it has an ease of access, you know, the readiness to be a part of a sample. So all of this will make it easier for you as a researcher to proceed with your research, okay? Because if you can imagine this with the previous one, okay, where you have to have a lot of information beforehand, okay, which is why convenience sampling is very easy to do. And the advantage are such that it's very low cost, okay, and it's extensively used. However, the disadvantage are the variability and bias cannot be measured or controlled, okay, basically because it's not randomized. So you cannot project the data beyond the sample, okay, so, and you cannot generalize as well the findings. So that is convenience. Next is judgmental sampling. Okay. In judgmental sampling, or also called purposive, your sample members are chosen only on the basis of your judgment. Okay, so what does that mean? So the easiest would be you already know. Okay, so for example, you are doing a survey on UPM students. Okay, and you know that right now in COVID, for example, let's just say that this is happening. So only one college is habitable. So everyone who is staying in UPM has to stay at this college. So in this case, your sampling, okay, although you are doing for the whole population, but you know that from your judgment, okay, choose only this college, people from this college. Okay, so that is your judgment. It's based on your judgment. So the advantage is that there is an assurance of quality response because you somehow through your judgment that these people are people that you can have access to, okay, rather than if you're looking at the whole of UPM, maybe some people you have to also consider people in Sarawak, for example, in Bintulu campus. So maybe people from Bintulu you cannot go, okay? So you might have problem if you are looking at the whole of UPM. So that's why you choose only one college, okay? So you can meet your objective, of course, but the disadvantage of using this type of sampling is, of course, again, bias selection, okay? It's a time-consuming process. I mean, it's basically for almost all type of sampling, and the potential inaccuracy in your criteria might result in problematic sample selection. Okay, so judgment sampling, it can be done, but you have to properly design it. Okay. Third one is snowball sampling. Snowball sampling is purely based on referrals, hence it's also called chain referral sampling method. Okay. So you imagine this is you, okay, so you interview one person, okay, regarding your research, what you are doing for your research, and then based on that, because you see that this person is very helpful, okay, it's very enthusiastic, so you ask that person, okay, do you know other people who might be interested in answering this survey? Okay, and that person will introduce two more. Okay, this is what happens here. So introduce to two other people, and then that will proceed. Okay, so these two people will introduce you to other people. Okay, so what is something that I want to highlight here, because a lot of people nowadays are using, actually you are using snowball sampling when you share the link. Okay, when you do your online survey and you share your link to people, okay, and that you ask them to also disseminate that. Okay, this is a type of snowball sampling as well. Okay, so with this technique, your sample number will increase in size. Okay, because imagine it will proceed according to how fast and how enthusiastic that person is in disseminating the survey. Okay, so you are the one who has to justify whether it is enough or not. So when to stop collecting, that is under your prerogative as a researcher. Okay, so that's the advantage is it's low cost, yes, and it's also useful in specific circumstances. Okay, and sometimes in locating rare populations, right? Because it's you might not have access, but the people you're talking to might. Okay, so that's why it's easy to do snowball sampling in this condition. However, the disadvantage is it's not independent. Okay, you are totally dependent on the people who are disseminating the sample. Okay, disseminating the survey. So projecting the data beyond the sample is not justifiable. Okay, because it's totally not random. Okay, so you cannot generalize. And basically, this is one of the things with non-probability sampling. Okay, so keep that in mind and make sure that when you are designing your sampling method, you take all of this into account. Right. Lastly is quota sampling. Right. So in quota sampling, you select your sample based on a standard, okay, that you already set in the beginning. So it's very, very similar to stratified sample. Okay. However, if you remember stratified sample before this, it's the selection of the elements of the sample are random. However, in quota sampling, it's entirely non-random. Okay. So you have, for example, here, your population, okay, a lot of different types of people, different gender, different age, and actually your quota is male above 50, okay. So you only focusing on that, and it's entirely non-random. So that is why it's called it falls under non-probability sampling, okay, because it's not random. So the advantage is that method of collecting samples, and you use this when you have limited budget, okay, because you don't need to prepare a strata list, which is what you have to do if you are doing stratified random sampling. Okay. However, the disadvantage again is variability and bias cannot be measured or controlled. It is quite time consuming. Okay. And lastly, of course, projecting data beyond sample are not justified. Okay. So from all of these sampling techniques, okay, there are basically two types of errors which can arise, okay, especially if you're doing surveys. So one is bias error, secondly is unbiased error, or called random sampling error. Okay, so in bias error is usually it's due to the selection of sampling techniques, or how you select a sample size. Okay, that will fall under bias error. Whereas for random, okay, it's basically differences between members of the population. Are they included or not included? Okay, so it's totally random. It's not something that is due to you as a researcher. Okay. So how to reduce sampling errors is, these are some of the tips. Okay, specific problem selection. You have to be very aware of your research problem, okay, and how you're going to choose the population based on this. Of course, you must also have systematic documentation of related research. Okay, so you can actually use the previous research methods as a basis. Okay. Enumeration, right, in samples, of course, a lot of the time people try to ensure that you have your your response rate is good enough by offering enumeration. Okay, and of course pre-testing, effective pre-testing is also very important. Okay, because you really want your questionnaire or your interview question to be very good enough to answer the questions in your research questions, basically. Right? And of course, the selection of sampling techniques. right? Which one of this, of all of this that you want to choose to proceed with your study. So you have to choose it very appropriately and be very careful. Okay. Whereas for non-sampling errors, okay, these are some of the cause, okay, that will lead to biases or mistakes in selecting the samples. Okay, so you will look into this further when you are developing your sampling method. Okay, but yes, just be aware of that, and then make sure that you try to include, you know, these possible issues. Okay, think about how to reduce that while you're developing your instrument. Okay, now we will look a little bit into determination of sampling number. Okay, so basically how many is enough? So this is a question that I heard a lot from my students as well, from people who are doing a lot of social sciences work. And also for science, for pure sciences, for those who are doing experiments, you rarely ask about sampling number, because we usually, the samples are already, you know, you choose it in a way that it will represent the whole population. Okay, but if you're looking at people, right, this is where it becomes blurry, because there's a lot of people are different from each other. Okay, so you need to think about how many is enough, how much number of your sample is enough. So the key point here is qualitative researchers, you seek saturation. Okay, it's not a matter of how many, okay, but what's more important is, have you learned enough? Okay, do you now understand about the phenomenon? So mere numbers are irrelevant. Okay, you want deeper understanding. So in quantitative researches, you seek statistical validity. Okay, it must be valid, right? So how can you generalize your findings to the population? Okay, have you excluded anyone? Why? Okay, so these are the questions that you ask. So the most basic thing that I can highlight is, for qualitative researchers, you do your sampling until you reach saturation. So saturation here means that the information that you get is enough, right? So a lot of, remember back about qualitative research, the type of data that you will get. So they are in the form of words. So when you say saturation, so enough, maybe around, you keep on doing your interviews until you see that, because when you do your data collection, okay, you will see that, oh, I found this word being mentioned time and time again until it's almost 90% of the people that you are going to see. Okay, so that is where you have found saturation. Okay, so that's why it's hard to say how many is enough in qualitative research, because you look at the saturation. Whereas for quantitative researchers, you already have your frame basically. Okay, and you want to know, is the cause and effect of these people, of this group of people, okay, does it apply to your hypothesis, okay, or is the null hypothesis true? Okay, and the way you can check that is by using your statistics. Okay, so from there you have to know already how much sampling number that you need to have. Okay, so that's the difference between qualitative and quantitative research. You know, the sampling numbers, how many is enough. So most probably, so for probability sampling, how you want to determine your sample size. So optimum sample size is required, okay, because of this. So you want to do appropriate analysis, you want to make sure that it's accurate, and you want to verify the significance, okay? Because always remember, in quantitative research, you want cause and effect, okay? You want to be sure that this issue or this treatment, okay, is related to the cause, all right? And you want to make sure of that using your statistics. Okay, so if your sample number is too small, okay, even if your study is very well conducted, the design is very well, it may fail to answer your research question because your sample is too small to represent the whole population. Okay, so in that case, you cannot detect any associations, or you cannot link the factors to your, so you have your independent and dependent variables, remember, so you cannot link that, okay, or it may be only associated in the case where there is a false positive, okay, when you're doing your analysis. But if the sample is too big, also a problem, okay, because your study will be very costly, very difficult, okay. You might have a problem with your time constraint, okay, and you may have limited number of samples, basically, so you lost your accuracy there. So you have to have an optimum sample size, okay, and the approach to estimate this is, usually you have to look at your study design, of course, and the main outcome, right? So the approach to calculate sample size, there are a lot of different ones, okay? If you go on Google and you search about your sample size, how do I estimate my sample size? Okay, a lot of things come out, right? Because basically there are distinct approaches for different study designs and outcome measures, which is why in this course, it's very, not only in this course, when you are doing your research, it's very important that you look at your study design, okay, and you decide from there how you are going to proceed. So at what stage can sample size be addressed? So you can actually do this two ways, okay, two ways to look at sample size. It's either before you conduct your experiment or your research, okay, and for this case, you're doing it in the planning stage, okay, or you can do post hoc, right? So after you've done your research, okay, these are for certain people who are starting the research without calculating the sample size. Okay, so you do that afterwards. So you want to see whether the sample number that you have is enough or not. Okay, so two stages, before or after. So you have to decide on that. So if you are looking at calculating it now, okay, before you're doing your research, okay, there are 4 procedures that could be used. You can use formula, you can use a ready-made table, okay, nomograms, and also computer software, right? So for formula, there's actually a lot of formulas for calculating sample size, okay, and power, because remember just now, different situations, different study designs has different types of formula. But what I'm sharing here is I mean, this is quite a general formula. A lot of people use this because it's for a population-based study with continuous data. Okay. So a lot of studies that you guys will probably do is related to this. Okay, so there are 3 requirements. You have to specify the amount of confidence you wish to have. Then you have to estimate the variance in the population. And then you specify the amount of desired accuracy. So what does this mean? Okay, so don't worry. This is the standard sample size formula. Okay, this is your sample size. So what you want to calculate, right? Z here, okay. This is related to statistics, okay, but don't be afraid of statistics. It's quite logical, okay. So Z here is, okay, remember just now I talked about normal distribution, okay. So normal distribution looks something like this. Okay, so the easiest one would be the age of population. Okay, so imagine the whole population of Malaysia. So a lot of people will fall under this. So imagine if this is infant, so 0. So maybe we'll cut that at 100 years old. Okay, so the large number of population will be at the central. Okay, I'm sure that you guys are aware of this. So Z here is when you say 95% confidence interval, is you are sure that around 95% will fall within this. Okay, so it's easy to look at that. So a lot of people who are living in a country, okay, not only Malaysia, this is like for world population as well. So the age falls between 0 to 100. Okay, of course, there's a lot of people who are, but for the sake of generalization, look at 0 to 100. So these are adults. Okay, so the amount of adults are usually the highest compared to infants and the elderly. So usually people will stick with 95% confidence interval when you are calculating. Okay, of course, there's also, if you want to be very specific, then you have to go for 99%. Okay, but 95% is very general. There's a lot of people are using that. Then P here is actually your variance. Okay, the estimated proportion of target population having a particular characteristic. Okay, so usually if you have no reasonable estimate, you use 0.5. So you might ask where you can find this. So this is basically if you are looking at previous research. Okay, this is where your literature review also come in hand, right? You can find out what is this from your previous research. Okay. But if there's no information at all, then you can stick with 50%. And Q is 1 minus P. Okay, so it's basically here. If you have this, then you will definitely be able to calculate that. And lastly is acceptable sample error. So by convention, it is plus minus 5%. Okay. But you know, if you want to be more accurate, for example, you will go lower, right? So why is this important? So the representative of this formula you can see here. Okay, so your accuracy, it will be less. Okay, sorry, the error will fall lower if you have bigger sample size. Okay, so look at that. I mean, here, if you have 2,000 sample size, you have an accuracy of 2.2%. Whereas if you have 1,000, you have around 3%, okay, sample size accuracy. And 5% here falls around here. So usually it's around 300 something. I can't remember. But yeah, 5%, 396 or something. So what is important here is that you want to know how many people, how many respondents is enough for your research. Okay, so if you have, imagine for this example, you have your target population with characteristic of 0.5. So this is your P. Okay, your Z statistics is 1.96. So this is available just now. So that correlates to 95% confidence interval. And then you want an accuracy at 0.05, which is 5% just now. So the sample size is 384. Okay, so this is enough to make sure that your the sample number, if you have 384 people, okay, answering and returning back your questionnaire, then you are good to go. So whatever findings that you get from that research is generalizable, okay, to the population, right, at 95% confidence interval with 5% plus minus. So basically, you might have something like, if we look at the previous example, the age, okay, so I'm just putting this there. So around, you find out that around 50 people, 50% of people are aware of your, if you're doing market survey, okay, for example, you want to introduce your brand. If you are a startup company and you want to introduce your brand. So you have 384 respondents, and around 50% say that, okay, I've heard of this brand. Okay, so your conclusion would be 50%, 50% of people are aware of the brand with plus minus 5% error. Okay, so around 45 to 55% of your respondents are aware of your brand. So that is the general conclusion that I can give you offhand. Okay, so in the second example, if your study population is less than 10,000, okay, and you want to basically, let's say you are doing number 2, because number 1 is already something, this research is already done, okay, by a previous researcher, and you want to repeat that with a different population. Okay, so let's say for this one is the target population is 10,000 or maybe 15,000. Okay, so it's more than 10,000. Whereas while you are doing your research, your population is less than 10,000. So you can use this formula. Okay, and after calculating, you find out that you need to have 286 people to be able to achieve the same validity of this one, okay, with a 95% confidence interval and 5% error. Okay, so this is if you are repeating, or if you are repeating a previous study. Okay, you are basing your research on a previous research. Okay, so this is where it's very useful. So that is through formula. There's also a ready-made table where you can use to calculate this sample size. So this is actually, you can find this in a lot of books, a lot of statistics books, research methodology books. So they have this ready-made table where you have your population size. Oops, sorry. You have your population size, okay, and your confidence level, and the error that you want. Okay, so you can choose from there. So if your population size is 1,000, for example, okay, I'll just highlight that very quickly. And you want to look at 95% confidence interval with 5%. So you have here 278. Okay, and you can just follow this, okay, to where your population is, right? So for example, if you have 100 people as your population, so you can look at here to achieve that. Okay, so this is 95%. There's also possibility of 99%. Okay, so you can use table, which is actually much easier. Okay, I did not go into nonogram because it's usually, I don't think a lot of you guys will use that. That is usually for clinical trials, so I won't touch upon that. But for number 4 is by using computer software. Okay, there's a lot of computer software that you can use to calculate your sample size. Okay, SPSS is there. There's also Stata. I'm not sure if you've ever used that, but it's very, very similar to SPSS. Okay. So with computer software, it's just going to be a matter of you punching in the numbers. It's very similar with the formula just now. So you have to specify what is your CI, your confidence interval, okay, 95% or 99%, and your variance, okay, and lastly your error, the margin of error that you want, okay, and you will get the number of samples. So that is all the 4 techniques. Okay, well, basically 3 techniques is if you're using that before you start your research. But however, you can also do this after you have finished your research. Okay, so if, for example, in somehow you cannot include the you cannot do the estimation for sample size. Okay, instead you will do power analysis after you have conducted the study. Okay, so basically this is with power analysis, it's related to your hypothesis. Okay, so if you remember, the probability of rejecting the null hypothesis is equal to 1 minus beta. This is actually power. Okay, it's basically like how you want to make sure that your null hypothesis can be rejected. Okay, so you want to make sure that your hypothesis is true. That is by rejecting the null hypothesis based on the statistical methods. Okay, so this might seem confusing, but remember, I've shared with you in week 7, if I'm not mistaken, a link to a YouTube video that is very, very good in explaining this power analysis. Okay, so you can actually look at that, because in the interest of time, we cannot cover everything. All right, so basically post hoc power analysis is done after a study has been carried out, okay, to help explain the results of a study, okay, in which no significant effects was found. So it can also be you know, if you're doing something, if you are not aware of how to calculate the sample number, for example, and you are already doing a research study, okay, so when people ask you, oh, how are you sure that your sample is enough? Okay, so you cannot do the previous one. You cannot do it research methodologies. Okay, so in power analysis, actually, you don't usually do it by hand. You use a software. So one of the most common software used is called G-Power. Okay, but it's also available in SPSS. Okay, it is one of the things that you can also use instead of other types of software if you are more familiar with SPSS. Okay, so basically it's just here. I mean, if you choose your power level, okay, remember this is done after you conduct your study. Okay, so if you choose 80% or 0.8, this is actually most common. So what you can see from the graph below, okay, if you have 0.8 here, you have N as 128. So if somehow before, while you're doing your study, you get 150 actually samples, okay. So you can safely say, okay, based on power analysis, the number of sample is enough. Okay, so you can, the findings of this research is representative of the population. So that is basically it. That's what you want to be sure of. Okay. Otherwise, if you have actually you have already done your study and you only get 100, for example. Okay. So, and the findings of the research says that you know, you cannot prove that your hypothesis is correct. So this is one way of explaining that. Okay, so you do your power analysis. You say that, okay, based on power analysis, the actual number is not enough. Okay, and which is why the findings of this research cannot prove that, you know, A is caused by B. However, you cannot simply say that this is not true because what, blah, blah, blah. So that is that forms your discussion while you're doing your analysis, you're doing your discussion based on your findings. So these are the things that you can discuss about. Okay, in power analysis is something that can be done. Okay, of course, lastly, if you are having higher power, okay, 95%, for example, okay, and you still have your 150 people, but you see here it's 210. Okay, so with 95%, you might not be, the power might not be enough, okay, but in 80%, it is enough. So it really depends on you how you are going to discuss this, right? So actually, we come to an end basically. So this next few slides, I leave it to you to study because these are actually important keywords, right? So I've already listed them here because while in this particular lecture, a lot of these things are very, very important. Okay, so look at that, study that on your own. So this will be, I hope it will give you a more understanding about some keywords that I've used in this lecture. Okay.